My name's Jonathan Oribello. I'm a Blue Marble Space Institute of Science contractor working as a bioinformatics programmer on the NASA GeneLab data processing team. Today I'll be talking about the GeneLab RNAseq consensus pipeline, specifically a Nextflow implementation. Before I get into the implementation, I want to give a quick overview of GeneLab. The GeneLab project seeks to accelerate space biology research through cataloging and democratizing space biology omics data. This includes curating rich experimental metadata, raw data, process data, all in one place. GeneLab also works to standardize processing approaches towards enabling multi-data set research. Ultimately, the work of GeneLab hopes to expand the accessibility of space biology data for research, all the way from citizen scientists to domain specialists. Today, I'm going to be presenting the Nextflow implementation of our RNA-seq consensus pipeline, which includes an associated staging, pre-processing workflow, as well as a validation and verification pipeline. A high-level overview of the consensus pipeline itself is as follows. 1, we take our raw FASTQ files, generate QC reports. Next, we perform adapter and quality trimming, as well as generating the associated QC reports for those trimmed reads. The star index is built and is used to align the trimmed reads to the genome. Before we quantify gene expression, we actually infer the strandedness of the original library using RSeq-QC. And then we combine the assessed strandedness, the star alignments, and an RSEM reference to generate our unnormalized gene counts. Those gene counts are then normalized before undergoing gene expression analysis with DSeq2, and additional gene annotations are added to generate a final annotated DG output table. I'll also be covering the two associated pipelines, staging and V&V, that work together with the core processing pipeline. Staging. Staging is going to be the pipeline that performs the setup before you actually get into processing. It includes two different routes for using the workflow. The first one is a fully automated processing for GeneLab datasets in particular, where you just have to provide the accession ID as well as a reference ensemble version that you'd like to use, as well as a route where you can supply a manually created CSV file for usage in processing non-GeneLab datasets. The V&V workflow is built on a framework written in Python. It runs after each major step and really has two major goals. First, keeping a detailed log of V&V checks on the raw and process data, including notable observations like outliers. And 2, to recognize egregious issues during processing like corrupt files, and halt processing, saving computational resources and allowing manual intervention. So here, I'm actually showing an example of the raw reads V&V, including the flag codes for each check, a portion of the checks that is output by the framework, and the output of a single check. So all of that will land in a final table for each check. Process data for the overall workflow is organized and is published for everything on GeneLab that is processed through this workflow. And these files are available for downstream research applications from the trimmed FASTQ files, as well as all of the alignment all the way through the final output table we get from the DG and GN annotation. In fact, I did want to showcase in particular an example of the kind of information you'd find in that final annotated DG output table. This is going to include plenty of mapped gene annotations, normalized counts for each sample, comparison statistics, dataset-wide statistics, and group statistics, all readily usable in downstream applications, things like GC or even just custom analyses. The code for this workflow is going to be on the NASA GitHub. but we are still currently working through some security release protocols. However, the actual processing steps themselves that make up the core workflow are documented there currently. Finally, I just want to note some acknowledgements. The GeneLab Analysis Working Group played a huge role in the design of this processing workflow, as well as GeneLab project funding that made the implementation with Nextflow possible, including the associated staging and V&V pipelines.